It is the chapter of MSc4th Sem Botany relating to the Agrobacterium tumefaciens, which is a plant pathogenic bacterium that contains a plasmid. The tumor-inducing or TI plasmid. It is the nature's genetic engineer for the plant transformation. The tumor gene engineering is processed or else they are removed. TDNA is introduced into the agrobacterium. The agrobacterium is then co-cultivated with suitable target cells, embryonic suspension of callus to enable the modified TDNA to integrate into these cells. The scientist is making approaches to discover, to develop something novel. After removal of agrobacterium, they transform cells or grown up into plants under selection. Agrobacterium tumefaciens is an alpha proteobacterium of rhizobiaceae. The crown gall is being shown and the constructs are being shown here. The sites left, right, TDNA, TI, plasmid, origin of replication, origin of virulence, and the region of virulence here, auxin cytokinin, or here to be participated and to transfer the genes. Genetic transformation. Agrobacterium tumefaciens strain LBA4404 contained plasmid PB121 binary vector. For example, the binary vector has two genes. Within the TDNA region of the gene construct, the binary vector PB121 has the background of PB and 19. For some example, discovery, application. It contained a scorable reporter gene GUS. GUS means beta glucuronidase, driven by a CAM V355 promoter and NOS terminator and a selectable marker gene NPTLL. Fuse between the NOS promoter and the NOS terminator encoding the gene for the enzyme neomycin. Phosphotransferase conferring kanamycin resistance. Kanamycin is very important here to participate for transformation. The reporter GS gene was used to assess the efficiency of transformation. The GS gene has an intron in the coding sequence. The coding sequence, it is a very long chain of coding the genes to be transformed from the side of the TI plasmid into the cell. which can express the GS activity in the plant cell, but not in the cell of Agrobacterium. So that is a tool, that is the TI plasmid. Case of the transformed tissues as well as the transformed plantlets were selected by using kanamycin. So it is also a tool. This gene construct was specifically designed for the development of genetic transformation protocol in some lentil. Protocols are different, and the biotechnological tool as the protocol is very specific in all examples to be used for the development of some specific kind of plant structure, including resistance and yielding and pathogenic benefits. So you see the constructs are here. For example, the sites are mentioned here to be participated. The genetic sequence, the sites are mentioned here. These are maneuvered as the biotechnological tool or tools to be processed to develop the new kind of cell and plant. Transfer of genetic information from Agrobacterium to plants. TDNA stably disintegrates into the plant genome and thus genetically transforms its target cells. TDNA can be engineered to carry virtually any sequence without affecting the bacterium's capacity to transform its host. Thus, Agrobacterium has been adopted as the preferred tool for the production of transgenic plants. With agronomically and scientifically important novel traits, whether yielding, resistance, pathogenic qualities, storage qualities, many such traits may be there. Cultivable traits may be there within the easy time frame, early than the natural varieties. Conditions, they are altered here. They are introduced here, incorporated here to develop new qualities. The process mechanism of the transfer and transformation. And their studies have revealed the roles of many bacterial and host proteins in the TDNA's journey. It is a very long journey that you have seen in the chapter. The chapter which is revealing the complicated site to be transformed. It is a tool for the generation of transgenic plants and will further enrich our knowledge of basic cellular functions and host pathogen interactions. That's all.